Okay, hello everyone, guys. Here, please listen to this video from top to bottom of this because I'm going to present and demonstrate how to check the total alkalinity in the onboard going in voyage, or you can use this technique for assessing the total alkalinity using the kit because for onboarding purpose you can't take many kind of pipette and burette, and these are the beautiful instrument, only a kit containing the syringe. You can't take burette to anywhere. You can take this syringe, only 0.1 to 10 millimolar plus H plus unit here, and you can use this syringe instead of the burette. And this kit is purchased from Merck. This is sorry, this is disappearing due to some watering. Okay, no problem. I explain what are the kit contains. Okay, so this kit contains basically three different solutions. You can see that. So these are the three different solutions. So R1 is the color indicator, that is a phenolphthalein solution, and this indicator is useful to measuring the carbonate ion found in the seawater. Okay, and this is R2, which is the bromoglycerol green indicator. So this indicator measures the bicarbonate ion in the seawater, and this is R3. So R3 is the titrant. So these both are indicator, and this R3 is an titrant. That is, it is a strong acid called H2SO4, hydrogen sulfuric acid, and this is the titrant for your seawater. So you are going to measuring the total alkalinity. So first, so what is the procedure? So what is the principle to do the titration of the seawater for measuring total alkalinity? So here you can measure so many kind of titration method, but you can't think that it is not a non-standard method. It is actually from the Merck et al. Method, and you can use it in the classic titration method in the laboratory, but it is the same kind of method, but there is a difference. What is that? You can take the long pipette and burette, but it is the only a small miniature size of titrant unit. So this syringe only takes the titrant, so instead of burette in the laboratory. And so for phenolphthalein and methyl bromoglycerol green, so you can use that. So I am going to demonstrate how to check the titration of total alkalinity. Okay, so there are three different kind of samples we collected from the seawater. So you can see that this is a island called Whale Island, that is in English term Hare Island. So this island is far away from the Rameshwaram, and this is the largest island, one among the 21 island in the Gulf of totally 21 island. So this is very large island, and there are including many mangroves over there, and so in this island there are coral reefs are highly highly developed. Massive corals including plate corals are here, but we are analyzing the water parameter analyzing for continuously 1 year, monthly sampling and bi-monthly fortnight sample. And here in initial year, that is Jan 2019, there was many corals are enriched in this island, but today the corals are not reducing, but many corals are going to die. And we saw that many plate corals are died in this island. So we can't predict what are the problem here happens, but we can check the essential nutrient of aragonite. So aragonite, one of the calcium carbonate ion, it is the only like food for coral reefs. So for measuring the health of coral reef ecosystem, it is essential to measuring the aragonite saturation state. We can't directly measure the aragonite saturation state, so we can indirectly from the derivation of CO2 system, so we have to analyze the total alkalinity. Okay. um, so for we have collected 3 different samples called the surface, middle and bottom. So we already analyzed the surface, and now I am demonstrating with this middle sample. Okay, so there are 3 different depths. Bottom is like 3 to 4 meters, and surface you can not a depth, it is a 0 surface, and middle is like 1 to 2 meter depth. Okay, so now I am going to demonstrate the measuring the total alkalinity. So you can take 5 ml of sample. So you can see the container only 10 ml capacity, but we can take only 5 ml of the sample. Okay, it is little bit higher than 5 ml, so we can pour back until the 5 ml comes. Okay, so we reaches 5 ml, and first it is the sampler, and we can add 5 ml of the seawater. Okay, so you can see the 5 ml is exactly in the utensil, and we can add, this is the phenolphthalein R1 solution. We can add 2 drops of phenolphthalein indicator. Okay, so you can see that it turns colorless from pink color, so which indicates the carbonate ion concentration is much more higher in this seawater. Okay, so now we can titrate it, which called strong acid H2SO4. It is sulfuric acid, so we can add some concentration of sulfuric acid. It is a diluent. Okay, so you can see there are a measurement, so 1 to 10 m millimolar per H plus, millimolar per liter of H plus ion. So we can using the syringe up to 0, so exactly 0. So you can see this exactly, it reaches 0. Okay, so we can take the titrant. Imagine it is like burette. It is classic method, no alteration, only the size is reduced. So, and we will add the titrant into the seawater sample of added phenolphthalein. Okay, now I will add up to the pink to colorless form develops. Okay, okay, I just add 1 drop. It turns pink color into again colorless, so that means total carbonate ion was consumed by strong acid. Okay, so now we can check how much titrant was consumed. Okay, you can see from 0 to point 0.32, 0 to 0.32. Okay, we can measure it, and we note it 0 to 0.32. Okay, and so this water is saturated with the carbonate ion. There is no carbonate ion which is consumed by titrant called H2SO4. Now we can calculate the carbonate ion concentration by this measurement using the different calculation. Then for the same solution we can calculate the bicarbonate ion. Now there is a indicator called bromoglycerol grain which measure the bicarbonate ion concentration. So again I added 2 drops of bicarbonate ion. So to the same solution I added 2 drops of bromoglycerol grain. Yeah, you see it turns green color. So after green color develops, we can titrate with the sulfuric acid. So first I add 1 by 1 2 drops, I shake it. There is no color change. So it has to be changed into red color so that the total bicarbonate ions are consumed by the titrant. So there is no red color. So I will again add the same H2SO4 titrant into the seawater sample. I add another 2 drops of sulfuric acid. Oh, there is no color develops. Okay, no color changes. I add another 1 drop. Oh, exactly. You can see the color develops into blue color. Okay, but it's not enough. We have to add other drops. Yeah, it turns red color, which indicates total bicarbonate ion is consumed by H2SO4. Okay, so you can measure the range from 0 to what? 2.5, 2.6, 2.7, 2.72. So totally 0 to 2.72. So we can note it down 2.72. Okay, so this is the difference between titration method from laboratory and on-board measurement. So you can't take many kind of instruments like burette, pipette, conical flask, beaker, and large amount of pack you need. So it is a very simplest method using the kit. You can check it very easily, and this is very helpful. I can check the measurement with using both laboratory method and Merck et al. Same kit titration. There is no more deviation. Okay, exactly those are same. Only 0.02 micromole per kg water concentration is vary, but it is more accurate. So I recommend that this method is enough for assessment of total alkalinity, and using the total alkalinity and along with the 3 different parameters called pH, temperature, and salinity. So we can measure the remaining 10 different parameters like PCO2, partial pressure of carbon dioxide, FCO2, fugic acidity of carbon dioxide, TCO2, total carbon dioxide, that is dissolved carbon dioxide in the seawater. And you can see the bicarbonate, carbonate from this method, and we can also see the boron alkalinity, calcite, aragonite, rivalry factor. Rivalry factor, you know that? Rivalry factor is the how seawater can able to get the carbon dioxide. So the ability of carbon dioxide getting is called rivalry factor. So we can take the 10 different parameter using the 4 pH, temperature, salinity, and total alkalinity. Using these 4, we can derive that another 10 different related seawater carbonate parameters using CO2 calc software. So you need CO2 calc software along with these 4 different measurement: pH, temperature, salinity, and total alkalinity. So we are checking the 4 parameters in 3 islands. This is one of the island, and another 2 island is far away from this island, which is about 8 to 10 kilometers away from this island. So we are measuring the 4 parameters from January 2019, and this is February 2020. So actually we are measuring 1 year plus 2 months. So 1 year plus 2 months we are taking the physico-chemical parameters of pH, temperature, salinity, and total alkalinity. So we have the huge data. So that is related 10 different parameters in our laboratory, in our dry lab. So we have more than 1 year data of aragonite saturation state. So actually there is no more publication of aragonite saturation state in India, particularly not in Gulf of Mannar. So we are the first analyze. We first analyzed the aragonite saturation state in Gulf of Mannar, and nearly our project called NICRA, ICER NICRA project will be ended on March 2020, and after that we are writing the paper and publishing the high reputed impact factor of open access journal. So very soon we will publish, and we will share the data to contribute to the world. Yeah, so those they are not unknown about the carbonate saturation state, and not able to predict the coral reef health. So very soon we will publish, and we will see. Okay, so thanks for paying attention to from top to bottom of the video. Thank you very much. Bye. So thank you very much for paying attention again. So I am Rangesh Kannan from Madurai Kamaraj University. I am working as a PhD research scholar and project fellow in ICER NICRA project. Actually, my PhD work is also ocean acidification research, and this NICRA research project is also assessing the coral reef health monitoring, and both are nearly assessing the ocean acidification. So for contribution to study of ocean acidification in India, we are very hardly working. Actually, you know one thing, we are coming from morning 5 a.m. So, and we are reaching the first island called Manoli around 6 to 7 a.m. And after that we go to Manoli Island by 8 a.m. and after that we come to Hare Island by 9 a.m. So 6 to 9 a.m. 3 hours we take for on board boat, and for 3 hours, morning 3 hours, and we will going to collect another evening sample from afternoon 3 to evening 6 p.m. So monthly once and fortnight samples we have collected in the island, in these three islands. So yearly we possibly will publish the data in a reputed journal of climate change oriented research article. Thank you. Bye bye. Yes, bye. Rangesh.